Here we have an operon called Lac Operon. This is commonly found in E. coli and is used in the metabolism of lactose. As you can see here, we have our wild strand of our Lac Operon, and all the parts of which are denoted by positives, meaning it's wild. As you can see here, let's start talking about the I region. The regulatory gene transcribes for our repressor, as seen here. Our repressor binds to our operator, denoted by a Lac O, right here. Our operator is preceded by a promoter. The promoter, denoted by a P region, is where the RNA polymerase will bind, and because it has the -35 and -10 regions. We also have, as seen here, our structural genes. These are the genes in which we're going to be transcribing and have our enzymes needed. As you can see here, there's 3 major ones in the Lac Operon: Lac Z, Lac Y, and Lac A. These code for beta-galactosidase, permease, and transcetylase. In the absence of lactose, however, the RNA polymerase cannot bind to the promoter, as the operator and repressor take up a portion and overlap with the promoter. This means there will be no transcription of these structural genes. Now, in the presence of lactose, as seen here with all these disaccharides, an inducer binds to the repressor, causing an allosteric change, enabling it to unbind from the operator. This allows RNA polymerase to bind to the promoter and begin transcription of our structural genes. As seen here, they transcribe. These transcribed enzymes are able to interact with lactose and create glucose and galactose. It's monosaccharides. Also, beta-galactosidase can interact with lactose and, in a positive feedback loop, create the allolactose used in the repressor. There are several mutations, though, that can happen in the Lac Operon, as seen in the chart right here. Let's talk about a couple in the example to the left. As you can see here, we're in the absence of lactose, and our genome has some mutations from a wild strand, as it has a super repressor. That means it cannot bind to allolactose, a structural mutation, that means it results in a defective enzyme, a constitutive operator, that means it cannot bind to a repressor, and another defective enzyme. As you can see here, in the absence of lactose, the repressor stays on. This would normally result in 0 enzymes, though there is another strand of DNA that can code for proteins. As we see here, we have a operator that is consecutive. This means, and a super repressor. The super repressor will try to interact with the operator, though it cannot, as it cannot bind. This means that RNA polymerase is free to transcribe, creating a functional permease.